DNA fingerprinting. Have you heard about DNA fingerprinting? And where did you hear? Where did you generally hear? Yes, very good. So generally in forensic departments, we will use the DNA fingerprinting or DNA profiling. So let's try to understand what is DNA fingerprinting. Okay, so best thing is that, so let's go for DNA fingerprinting. If we talk about this, let's say number maybe. So DNA fingerprinting is known by other names. So it is also known as DNA profiling. So this is one name. In forensic department, usually this method will be used. So in simple fashion to say what is this, it is a technique. So it is a technique, generally it would be used for what? To identify genetic composition of an individual. So this would be the basic technique, DNA profile. So here, what we will do and what is the principle that we will use here means, if you look from the genetic composition of humans, around, if you talk from the principal point of view, around 99.9% of the DNA of humans is similar. Only 0.1% of the DNA of humans varies from individual to individual. And this varying DNA is the base for this identification of the individuals. That means DNA profiling is a technique that is used to identify the individuals in certain situations. Okay, so from a pool of given samples, we can identify particular individual. Okay, so principle is this. So this slight variation with respect to genetic composition would be the theme. So here, what is the procedure that we will use here? Okay, what is the procedure we will use? So first, what we will do, whenever we will get, so whenever we will get a sample, so we will have a sample. So from this sample, what we will do, we will try to separate the DNA from the sample. Okay, so we will separate the sample DNA, we will separate DNA from the sample. Okay, then after separation, it would be a single copy. So in order to study or in order to properly identify the genetic composition, we need to amplify the DNA sample. So for that process, what we will do, we will try to multiply the DNA. We will try to multiply the DNA through the process. So what process would be used for multiplication of the DNA means, it is nothing but PCR. Okay, polymerase chain reaction. Polymerase chain reaction is the technique that is used to multiply the separated DNA fragments into more number. Okay, so when we get more number of DNA samples, then we can assess the things properly. So PCR, polymerase chain reaction, is the process that is used here. Okay, so here what we'll be doing for making multiple copies of the DNA, we are using PCR. So as we are producing multiple copies of the DNA, so the process, PCR process, it is also known as molecular photocopying. Molecular photocopying. So have you heard of, we have already learned about molecular scissors, molecular glue, and now this is molecular photocopy, because we are producing copies of DNA. That means at the molecular level we are doing it. So that is why it is called as molecular photocopying. Okay, so now with the help of PCR, we have multiplied the DNA, and after multiplication, then what we will do, then we will try to arrange these DNA into visible bands. Okay, so we will arrange them into the form of visible bands. So let's say, so they will have that arrangement. So it would be like a kind of, assume like, remember it would be like a kind of barcode, so which will contain some visible bands. So based on that, they will get that profile. So then they will match up. So likewise, so for segregating them into a form of visible bands, based upon their the colors, all those things, they will do that. So for this process, they will use another method called gel electrophoresis. Gel electrophoresis is the method that is used to make this multiple copies of DNA into visible bands. Okay, so there what they will do, they will drop all these DNA fragments into a gel based suspension. So there, based upon and then they will subject this gel for electrocution, means like they will supply electric things to this. So then due to that electric effect, so all the DNA fragments would be arranged into a form of bands, visible bands. Okay, so that process is gel electrophoresis. So they will use this thing. So after when they become visible bands, then they will get complete band patterns, of DNA. So thus they will get the profile. Profile would be decoded, and in the similar fashion, they will take the thing of suspects. So then they will match it. So whichever matches, so that becomes like this. Okay. So generally, what you have to remember here is that you have to remember that in DNA fingerprinting process, one thing like PCR technique would be used, gel electrophoresis technique would be used. So PCR technique would be generally used to multiply, augment the number of DNA fragments, and gel electrophoresis technique would be used to, it is used to segregate the DNA into visible bands, so that they will get certain band patterns of DNA material, so that they can have a profile.